First, I'd like to acknowledge and celebrate the first Australians on whose traditional lands we meet today, and to pay our respect to the Ngunnawal elders past and present. So today we have Associate Professor Naomi Hammond, who is the Critical Care Program Head at the George Institute of Global Health, with a conjoint appointment at UNSW, and she also works part-time as an intensive care clinical research manager at the Royal North Shore Hospital in Sydney. So Naomi's research interests lie in sepsis, ward resuscitation and fever management, as well as post-critical care outcomes. As a critical nurse researcher, Naomi has led a program of sepsis research, including facilitating 4 clinical trials during COVID-19 in India. She leads the Australian and New Zealand Intensive Care Society Point Prevalence Program and has also undertaken international point prevalence studies. Naomi is a NHMRC Emerging Leadership Fellow and has so many hats and appointments. I won't go into them today, but I will point out she was just nominated for the New South Wales Public Servant of the Year for her contribution to critical care research and improving patient outcomes. And welcome, Naomi.
Thank you very much for that introduction. It's an absolute pleasure to be here today, and I also would like to acknowledge the traditional owners of the lands of where we're meeting and pay my respects to elders past, present and emerging, and welcome any Aboriginal and Torres Strait Islander people joining us today. And just acknowledge this beautiful artwork by one of our colleagues at the George Institute, who's part of our Aboriginal health program, the Gunnamana program, which means heal, and we love to start with this. It's a beautiful piece of work. And so today, when Lizzie invited me, there we go. Lizzie invited me and she said that they've started a new speaker series here around clinical trials. While I understand that many of you may not participate in clinical trials, she said it is of interest to this group to understand a little bit about the background and history and how we go about doing trials. And so I thought, well, I've got a great story to tell you about clinical trials in critical care, and Imogen's here and she would know the story very well. It's a story of intrigue and drama, but mostly blood, sweat and tears. But that's been conducted by my mentors and many of the intensive care clinicians around Australia and New Zealand over the last 25 years. And so what I'll talk today about is, I didn't think many of you would know what intensive care was, so maybe just a little brief note on that, about how we built a culture of research within the intensive care environment, about some of the research institutes that we partner with to operationalise our trials, the challenges that we've faced, the future directions of where our intensive care trials are going, and then some of the impact that we've had. And so a bit about me, as Lizzie explained, I do sit between a couple of organisations, one being Royal North Shore Hospital, which is a large tertiary referral hospital in the northern parts of Sydney. It's one of the main referrals possible for burns and spinal cord injuries. There's the neonatal intensive care unit, and where I work is the adult intensive care unit. And that unit has about 58 beds, up and down depending on money and nursing availability, but 2 general intensive care units where we have patients admitted. That's where we have our burns patients, but patients who may have suffered from trauma, those with sepsis and some other conditions. We also, the other 2 units are a neuro intensive care unit and a cardiovascular unit. And then my other place of work is at the George Institute for Global Health. This is a medical research institute that has offices in Australia, China, India and the UK. And you can see there's quite a large number of programs of work that the George does. But their main mission is really to take therapies and trials to lower middle income countries and try and improve outcomes for others who may not have the resources that we do. And so from an intensive care perspective, I've got this lovely image on the left from like a stock image online. It's very nice and clean and calm, whereas the reality is really on the other side, where if you're in intensive care, I hope none of you have experienced that. I know there's a neuroscience group here, so maybe you are quite familiar with this environment, but it's noisy, It can, many people coming through, if you're awake as a patient, it can be quite overwhelming. It's pretty overwhelming for for family members as well. And you can see there's a lot of machines and lines and just stuff going on. It's quite a busy environment. And from a clinical perspective, each day that you're with patients and making decisions, the decisions that you make may and likely have huge impacts on patient outcomes. And when I started out as an intensive care nurse back in the early 2000s, the common treatments that we were giving to patients in the intensive care unit consisted of having tight glycemic control, so giving high doses of insulin, using high doses of steroids. We were sedating patients heavily, and we were using a lot of intravenous fluids for resuscitation and other for fluid therapy. And really, a lot of these interventions that we were doing didn't have true robust evidence to back why we were doing these therapies. And so this is where we embarked on the journey around the intravenous fluid research. And for background, I don't know how many of you are aware of different types of fluids. So there's usually categorized into two types, crystalloids and colloids. Crystalloids you'd most likely be familiar with in respects to normal saline. And then there's other types of crystalloids called balanced salt solutions. And these fluids are easy to make, they're cheap, readily available, they have small molecules that are meant to go into the intravascular space and expand blood volume, but potentially some of the downsides is that it might leak into the interstitium and cause swelling and edema. And then for colloids, the thought is that they're perhaps better for the use of increasing intravascular volume. They're larger molecules meant to stay within the intravascular space, but the majority are synthetic, other than human albumin, which is produced in Australia as part of the blood fractionation process and is readily available to intensive care units to use for fluid resuscitation therapy. And so back in the late 90s, a Cochrane review came out around human albumin. And this was quite a concern for our group in intensive care, where we were using a lot of albumin to resuscitate patients. And what this Cochrane review showed for the hypothermia trials that were included, there was only a small number of patients studied, but that there was a suggestion of a 6% increase in death. So this was saying around 3,000 unnecessary deaths in Australia each year. And so the conclusion from that trial, from that review, was really that albumin should be halted outside of large-scale trials. And so this caused huge drama and news around Australia, because human albumin was used a lot, and it was the treatment used in burns quite most of the time. And so the concern was, well, should we be stopping this this treatment that we're using readily? And so what our group did, my mentors, Professor Simon Finkel, Professor John Myberg, and others, looked at that, well, there's an opportunity here to conduct a trial, but how do we actually do that? We've never done a trial of intensive care. So what do? We know that there was a lot of people receiving the intervention. Hospital mortality was sitting around 15% at that time. Clinicians were interested to know, well, is this actually safe to use in my practice? And we felt that there was an ethical imperative to really find out if this current practice was actually causing harm to our patients. And so Simon was working at Royal North Shore Hospital, and what he was told was that there was a new research institute that had just opened up down the way in a building down the road called the Institute for International Health, which is now the Jordan Global Health. And so he went to Stephen McMahon, who had he was a public health epidemiologist, researcher, who had started this institute with his wife, Robin Norton, who was again a world-class epidemiologist from the UK. And they talked Simon through, well, here's what you can do. And so what did they need to conduct a high quality trial? We know that they needed people, they needed steering committees, management committees, data managers, operational staff, particularly, most importantly, as the project manager, DSMB statisticians, and outcome adjudicators if required. And the George Institute was able to offer this to the group, and they also were able to offer all the high quality documentation that you're required to have to conduct any type of trial. And I put the picture there of the electronic case report form. I know that's very standard these days, but this was not standard back in 2000. We were using paper-based forms. So this was a luxury really at that time to have available. And the other need was to have intensive care units and to have intensive care units interested in randomising patients to this potential trial. And what had just started up only a couple of years before was the Australian and New Zealand Intensive Care Society Clinical Trials Group. And this group was a group of interested clinicians who wanted to conduct research. So they got together and came up with ideas of what they could do, what research questions they had in intensive care. And one of the first big trials, I don't know whether it was Lancet worthy, but it got in the Lancet. It was pretty amazing for this group. Was conducted only just before this issue around albumin came up. And so what it did, the dopamine trial was in 23 ICUs across Australia, small number of patients. They had research coordinators or nurses really at the bedside collecting data. There was no formal structure to that. But what it did was build a culture and a collaboration to be able to conduct large-scale trials. And the other thing to do a trial using albumin, hopefully a blinded trial, which is what the SAFE study was, the saline versus albumin trial, was to partner with industry. So have the industry be able to produce a blinded product, so both your placebo or if you had a placebo and the intervention product. And so we partnered with CSL Behring, where they were able to produce both the albumin, which they were already producing, but also put saline into identical bottles. So if we think about when we conduct studies, blinding is always a huge challenge, and whether it's actually oral tablets, probably one of the most challenging products to blind. But for fluids, I've just shown you a couple of examples there from the trials I'll talk about. But the SAFE trial, which was albumin versus saline, albumin actually has a yellow colour to it, so it's very obvious. So it looks like, well, how do you actually blind this to saline, which is clear? And what they came up with there, you can see the little blue or green colour and the giving set, and it was kind of covered so you couldn't see the fluid, and then that was the technique to blind the trial. You couldn't see whether it was yellow or not. And then consent. So consent in patients who lack capacity is obviously quite challenging, and there's different regulatory processes around the different states and territories of Australia. And the Guardianship Act actually hadn't really taken into account clinical trials, and it was amended to account for clinical trials, but it still stated that to conduct a trial you must have ethics approval, but also informed consent has to be achieved before including someone in the trial. And so this was incredibly challenging, obviously, in an intensive care environment or an emergency setting where you can't, patients are unable to consent for themselves. And so Professor Simon Finkel actually wrote to the Guardianship Tribunal in New South Wales to say, well, here's what our trial is. We can't, we're looking at 7,000 patients to recruit. Is this a clinical trial in respect to the Guardianship Act? And they reviewed the documents and determined that SAFE was not a clinical trial in the context of the Act, where we were using established therapies within the clinical setting. And so this enabled us to conduct a trial where you could include patients up front and then get what we're calling deferred consent down the line, or now consent to continue is more common term.
If you've done that in Queensland or ACT, would you put the same?
So they each have different requirements. So Queensland you can register, you go to QCAT and just register the trial. If it's 2 standard therapies, that's okay. And then ACT usually follow the other states as to if we get approval in Melbourne, Victoria, where they actually approve waiver of consent, and then ACT potentially will follow that, or if it's in New South Wales, they'll go by what the NCAT has.
I know it's been different to do trials on emergency and other things at ACT because of the way they do consent for people that are not able to give their own consent.
That's right, yeah, and it goes through so trauma trials in the trauma setting as well, so it's quite challenging. And then to tell the story around these challenges in intensive care from a consent perspective, in 2015 the New South Wales Guardianship Tribunal actually changed their definition again, that was based around randomisation. And so this, to diverge from the fluid story, actually caused a major concern for trials that were actually currently running in Australia, but mostly in New South Wales, which is usually the biggest contributor to our trials. And so what happened, there was a new trial that was getting approval, looking at sedation practices called SPICE, and the NCAT ruled that this trial needed prior consent, and this was based on the methods of randomisation, but also the drug licensing of these sedative that they were using, which was dexmedetomidine. And this decision by NCAT around SPICE about having to have prior consent, which is particularly challenging so either the next of kin has to consent within a very short time frame. For SPICE, the inclusion window was, I think, about 6 hours. So your people are in a very difficult situation to try and to go to them to ask to consent to something that is standard practice is almost unethical. But then because of this decision, there were other trials that were running that had to be halted and suspended and had to go back to this NCAT to confirm, is it okay that we're including patients without a prior consent? And so these are some of the challenges that we've had to go through and navigate in intensive care running the trials. And so going back to SAFE, this saline versus albumin for fluid resuscitation trial, we were able to complete the trial, just under 7,000 patients across 16 intensive care units. We had the blinded fluid, it was successful blinding, and what the trial showed was there was no difference between the 2 types of fluids on mortality at day 28. And an editorial that was published at the same time with the SAFE trial really highlighted around how important this trial was for intensive care medicine, in that it was a landmark trial, but it was a milestone for the discipline in that it provided one of the first robust evidence for this group. And interestingly, the SAFE trial's secondary work from that, looking at patients with traumatic brain injury and those with sepsis, actually saw divergent results in mortality for those groups. But what SAFE did for Australia and our intensive care clinical trials group was really provide an opportunity and a culture change for intensive care research, and it really set the momentum and standards for ICU trials going forward. And again, it made the news. I love the headline, blood smear removed, but it truly was a big deal for intensive care clinicians that they were able to give either albumin or saline for their patients. And obviously using saline is cheaper than albumin, so that was a huge cost saving to the health system if people change practice. And what it did, it also established this operational expertise within the intensive care units. And so what it did is built what we call research coordinators in the hospital, something that I started out doing myself. And this network of research coordinators, we've put ourselves IRSI, so Intensive Care Research Coordinator Interest Group, started out as a handful of nurses working as research coordinators, and has now expanded across Australia and New Zealand to be quite a large group of successful operational individuals. And many of us have gone on to lead and undertake higher degree, lead our own research programs and undertake higher degrees, which I think is a testament to the encouragement from the ANZX group, and an important aspect to take home, I think, when you're running large research programs, to make sure you're succession planning for all that work with you. And it really led to this fluid, the SAFE trial led to a highly impactful program of work. And so I've highlighted, this is a timeline looking from the Cochrane Album and Review to current day. And this is the red highlighted boxes. On the top part of the line are the work that we've done. So the SAFE trial, I'll talk about the CHEST trial and then the PLUS study. But on the way, we also undertook, as Lizzie mentioned from my bio, cross-sectional studies. So we called them TRIPS work, so Translating Research into Practice Studies, where we both looked on a regular basis at clinical practice in Australia and New Zealand, but also internationally, to see how practice had changed over time. And so this work of looking at translating, looking at changes in clinical practice over time, highlighted a few changes in practice, not exactly what we thought we would see. But in 2007, after SAFE had already been finished, we saw that globally across the units that we surveyed, that colloids were the most commonly used fluid still, but the colloid that was most commonly used was hydroxyethyl starch. And this fluid had not yet made its way to Australia, and it was coming. So the thought was, well, there's only a little bit of evidence around this product that we're about to get in Australia. So perhaps we should be conducting a large-scale trial to determine the safety and efficacy of this fluid. And so again, we went through working with the George Institute for Global Health as the operational experts, able to drive and deliver on a clinical trial that again was blinded. We worked with Fresenius Kabi at this time to produce the blinded fluid, both saline, so 0.9% sodium chloride, and hydroxyethyl starch. It was 7,000 patients across 32 intensive care units. Now, during the first year of recruitment to this trial, we found out that a lot of the majority, actually, if not all of the evidence base for the rationale of why hydroxyethyl starch was being used, mostly in anaesthetics, but across the world in intensive care as well, was actually based on fraudulent evidence. So a German anaesthetist, Jacqueline Bolt, had led a large number of the small trials showing efficacy of the use of hydroxyethyl starch. But this data was found to not have ethics approval. A lot of the data was made up and fraudulent. So all this data was being retracted. But what do you do as a trials group running a clinical trial using a fluid that perhaps is maybe not should not be used in humans? And the decision was, went to the DSMB, went back to ethics, and all groups decided that, well, we need some evidence this product is being used in practice. We need to have evidence of whether we should be using it or not. And so whilst we were conducting our trial, the CHEST trial, the middle one, there was also a Scandinavian group, clinical trials group, asking a similar question, but specifically in patients with sepsis, and so not all intensive care unit patients. And both these trials were completed. The 6S trial showed increased mortality with hydroxyethyl starch and increased requirements for renal replacement therapy. And the Australian trial, while it didn't show a difference in mortality, it did show an increase in adverse events and need for renal replacement therapy. And all this data was put into a Cochrane review and essentially recommended that hydroxyethyl starch should not be used in clinical practice. There was also some meta-analyses that came out where they included all the bulk trials, but also those without, and did some sensitivity analyses just to look at trials that excluded the bulk articles. And there was a definite summary of evidence that increased in mortality associated with the use of hydroxyethyl starch. And so this was big news, and regulatory bodies took action, which is obviously great to see. And what we saw across the UK, the US, Europe, that there were changes to labelling, including either in the UK, the product was removed, but in many other places there was a black box warning added to the product to ensure people knew the risks in critically ill patients. Interestingly, if you look at sales of HES in the EU, this is after, so you can see where CHEST and SUCCESS were published, and then when the regulatory action took place, that there was a major drop in the use of the product there. But that wasn't without a lot of backlash from the company that we partnered with, Fresenius Kabi, and I was a PhD student working in this space, and I had a lot of legal letters requesting information from my PhD through the Freedom of Information Act in an attempt to get the CHEST data sent to them. And so the decision was made to reanalyse the CHEST data, but have an independent group conduct that. And even though that was done, still in the BMJ, you can see there was concerns that our independent reanalysis was perhaps not so independent. So the backlash from this trial, even though we would say as a group that it's definitely one of our major wins of removing a product that shouldn't be used, perhaps is thought differently to other groups. And unfortunately, sales of hydroxyethyl starch is still quite high in other countries, particularly in lower middle income countries. And this is data from China. I was recently, it was last year in India, at an intensive care conference there, and the marketing for HES was all around the conference. So it's definitely a product that can be still accessed if people choose. And so it just brings to mind some other ethical concerns around marketing back in the day, I guess. So going back to the fluid story and the choice, so looking at our translating research into practice work, after the CHEST study had been published and it was safe in 2014, we were looking at clinical practice again. And what we now saw was that there really was a shift towards crystalloids, and where we thought that saline had the most robust evidence behind it with SAFE and CHEST. We're actually seeing that people are using what's called the balanced salt solutions, which are more physiological, considered more physiological. And so there'd been this move towards those, but there was no high quality evidence to actually state whether these were best fluids to be used. And so again, we've gone down this path a couple of times, but we're like, okay, so let's undertake the PLUS study, the plasma light versus saline trial, to really gather some high quality evidence for the use of balanced salt solutions. And in this case, we use plasma light. And again, we partnered with Baxter, which was an excellent partnership. We've done quite a lot of work with them before, and they produced the blinded fluid for us. This was conducted across 53 ICUs, and we planned for 8,800 patients. And so here comes more of the drama. Oh, Sue, sorry. This was all the good bits. So we're running through our trial recruiting. We pre-published our protocol and our statistical analysis plan, which is considered to be the best way of doing this. So you're putting all your analysis plans out prior to closing your database. But then there was new data published as we were recruiting. And so what do you do? This new trial came out of the US from a single centre. It was a large centre. They were recruiting a lot of patients. They had 15,000 patients included in this trial. It was a cluster randomised trial, but asking the same question that our one was as well. So we kind of went through this. Well, does it unequivocally answer the question that we're studying as well? Do we need to stop or alter what we're doing? Do we need to inform the DSMC or and ethics? And do we need to change our PICs to let patients know that there is actually evidence out there on this question? And this trial, I'll back to the next slide, but it didn't, while it didn't show a mortality difference, but it did show differences in acute kidney injury. And so we were concerned about what this data may have shown. And so we did go to the DSMC and ethics. And while the trial had shown some harm, the thoughts were that we were looking at day 90 mortality, that at day 30 and day 60, there was no mortality difference. So by day 90, what would the outcomes be? And working with ethics and the Data Safety Monitoring Committee, it was confirmed that we should progress with completing recruitment for the PLUS study. But then COVID hit. And this made it incredibly difficult to recruit patients. For all of our efforts were going towards recruiting to to COVID trials. So all the non-COVID trials really stopped recruitment. And so the decision was made in late 2020 by the blinded study committee to actually stop recruitment at the end of that year. And looking at where our recruitment rate was sitting, we estimated that we'd have around 5,000 patients. And we knew from our power calculation, our mortality that we had, our combined mortality was sitting around the 23%. So we thought, well, we know that there's, looking at those power calculations, that at 4,900, we should be able to have enough power to detect a 3% difference in mortality between the two fluid groups. And we also undertook a post hoc analysis of whether data across the futility line or efficacy boundary. So just talking about recruitment challenges, I think this is probably one of the biggest hurdles in clinical trials. I've got the graph of our first trial, SAFE. So that is very rare to see where you've got actual recruitment higher than your target recruitment. And this, we love this slide because we wish this is what we would have all the time, but actually no. And the many reasons behind this, but probably the main one is that that SAFE was done on its own. It had never been done before. So everyone was invested in, interested and able to put the time in. Whereas with PLUS, you can see this is actually much more common where your target, your actual recruitment is well below your target recruitment. And if we're looking at critical care trials that were happening over that period, I put a line there for PLUS. It was one of many trials that was happening across the different research institutes that run intensive care trials. And there was major, in our intensive care research coordinating network, the growth in that network was still, it hadn't occurred. So we were still sitting at the similar numbers that we were early on in 2012, even though we're getting a lot more trials coming through. So this is where co-enrollment is important, particularly in our comparative effectiveness trials. SAFE, you can see there, sits out on its own about the numbers of patients recruited per month per site, but the majority of recruitment sits much lower. And so across our network, we each management committee actually talks to each other around, can we co-enroll into each other's trials to increase enrolment and ensure everyone can make recruitment. And patients, actually we've done a lot of work around consent and whether consumers and patients mind being asked to be included in multiple trials. And the majority of the time, patients say they don't mind, they just want to get better. So going back to PLUS, so the post hoc futility futility analysis. When we did this, we were actually pleased to see that we crossed the futility boundary at around 56% of our originally intended sample size. So if we had recruited to 8,800, really we would have seen no change in our results and no benefit. And so this work was published in the New England, and again, we found no difference between the two types of fluids. The 90-day mortality between both were the same in all secondary outcomes. And what we've been doing is, more commonly now, is undertaking systematic reviews and meta-analyses and co-publishing them with the main trials. And I would highly encourage people to do this if you're in this situation, to pre-plan. It takes about a year prior to get your systematic review protocol done and start doing the processes. But it's a highly effective way of giving clinicians that summary of the evidence, so both the new trial data, but also the summary of all the data together. And so this is something we've been undertaking. And also we had a pre-planned individual patient data and meta-analyses. So this was that group that had published before us. We were already talking to them about, let's pull our data and undertake an individual trial data meta-analyses. The other piece of work that we do now as well, we really try and build in long-term outcomes, economic analyses, and be part of guideline groups to really continue to provide as much evidence for clinicians to make an informed decision at the bedside. And today, the intensive care clinical trials group is one of the most successful clinical trials group. And here I've just listed the New England papers that have come out from the group. There's around 31 papers, and then there's others in JAMA and Lancet. We don't do Nature. I'm sure you guys are more Nature, but we'd love to be able to publish in Nature, but we're not doing that type of work. But it really just wanted to highlight the successes of this group, where it's driven by clinician inquiry and decision making, and to produce evidence to inform practice. And if we go back to my earlier slides about the common practice when I started, well, we've now seen major changes in these therapies and how we deliver treatments based on the clinical trials that we've conducted through the clinical trials group. And so where's the Australian New Zealand Intensive Care Society and the clinical trials of critical care going? We've started to really look at embedding research into practice and having a learning healthcare system where data is, we're able to pull data from our national registry and pull that into our clinical trial databases. So these are registry-based trials, but also having the platform. So our ANZICS registry has been around for quite some time, but it is a registry. So, but also building infrastructure to house our clinical trial case report forms within that setting. So it's all embedded within one area. We're also going down the path of platform trials, and I know this is pretty, it's not novel anymore, particularly with COVID, and many of you would have heard of REMAP CAP, which is the critically ill COVID platform trial, but it's definitely the way to go for trials, even though it is operationally extremely challenging to do. But it is the way of the future. And I think for critical care and all the different decisions that are happening at one time at the bedside for patients, platforms are actually the perfect tool to use, where you really do have multiple decisions at one time in the clinical setting. So it really emulates current practice. But what we'd like to see is actually building in the having a biological sample collection, which I don't think we do very well within our intensive care trials. And so we've proposed a sepsis platform, and this is something that we're hoping to get off the ground and get some funding for. It's very much led by our consumer group through Sepsis Australia, and partnering with both industry and other research institutes to really make this something that is appealing, I guess, to more than just a small group of ICUs. We're hoping to get all on board across the nation. And again, domains, the albumin story we feel has not finished, and I'm looking at imaging to say, and the crystalloid stories and steroids. So we're looking at these domains. Now, one of our areas that in intensive care we may not have been so good at is having consumer engagement and advocacy. And this is an area that we're really working on quite a lot. And I'm being more broadly from an intensive care setting, whereas with patients who experience sepsis, which is how the body responds to an infection, and it can injure your own tissues and organs and have you end up with multi-organ failure and potentially in the intensive care unit. Or for some people, you may not go to the intensive care, you might be on the ward. So the sepsis group have come together themselves as consumers in a large group online on Facebook. And they approached, again, my mentor, Simon Finkel, about starting up a network for sepsis. And Simon was really pushing to get, it was part of a global sepsis network, and really pushing to have a sepsis national action plan in Australia. And so this group, which we've now renamed to Sepsis Australia, has been extremely active, and a group that is really driving a lot of the research that we're doing today in our group at the George Institute. And they're co-designing programs of work. And one of the major pieces that have come out of the sepsis program is actually the George Institute and Sepsis Australia partnering with the Australian Commission and developing a sepsis clinical care standard. So this is completely divergent from clinical trials, but what we've found is actually this is almost like a priority setting for our consumers and for our group to really understand what is it what are the needs of our consumers in the sepsis space. And one of the pieces of work that I'm leading, which is not a trial, but it was really driven again by the consumers, and it's around what supports and models of care do we offer for people after sepsis. And the answer is we don't. And intensive care follow-up clinics are maybe the closest that we get. So we're undertaking a program of work to really drive this question and provide something for our consumers that they think is the priority for them. And I agree with them. And so with our George Institute program, so it's ever expanding, and the work that we're doing in the clinical trial space is obviously always there, but some of the newer initiatives are really trying to, we've got all these large data sets that we use, which are hugely rich and should be accessed. So improving our data sharing, improving our biobanking, and also bringing in implementation and process evaluation into our broader program of work. And so really to start closing, the key reasons for some of the successes for clinical trials in critical care is around the common goal that we've had to improve the outcomes for patients. And this is really driven by clinician researchers who really wanted to provide best practice to their patients. There's been a collaborative, cooperative, collegial group. There's operational excellence with our research institutes and our research staff, both within a program management perspective, but also within the intensive care units themselves. And it really is around prioritising the group over the individual and working hard, being passionate about what you want to do. Everything about science is fun. I don't know why I wrote that, because it's obvious, right? And having a culture of questioning and embracing new ideas and welcoming and supporting new researchers. And importantly, our trials have been looked at by the Australian Commission to determine have they actually been worth the money that is put into them. And we found that for every dollar invested in a clinical trial has returned close to $5 back into the health system. So we would say that they are, it's worth the investment in clinical trials. And just looking at some of the individual trials, the fluid trials that we've done for SAFE, the cost difference between albumin and saline was 16 million per annum, affecting 42,000 patients per year. For the secondary analyses in TBI patients, the cost benefit there was huge at 82 million and related to reducing deaths from the use to not use albumin in these patients. And then the chest trial, again, you can see the cost benefit there. So we do know that we have, by undertaking these clinical trials, we have saved thousands of lives, and we also are saving millions of dollars, which obviously the health system likes. So thank you very much for listening.